Κι άλλο λίγο. Εκεί λίγο ακόμα. Έτσι. Κι άλλο κάτω. Κάτω κατέβασε την. Κάτω κατέβασε. Κάτω, όχι το μου κόβει το κεφάλι. Κάτω κατέβασε την. Εκεί. Αυτός. Okay. So, from what we said up to now, any questions or comments? No. All right. So, let's now go into more detail. Let's start thinking about the different roles played by renal blood flow. Renal blood flow is used to sustain filtration and elimination of metabolic end products, we said. Όχι, ναι, βέβαια. Okay? Now, the other thing that the renal blood flow is used for is to achieve rapid changes in body fluid volumes and also in the composition of those volumes. And this can be done only through one way, by renal excretion of water and solutes. You just have to put out of your body what you have extra. The third reason that we need the renal blood flow as a, let's say, as a factor that determines our internal stability is to have it serve as a hemodynamic reserve. You have to keep in mind that the blood flow through the kidneys is about 1 liter per minute. However, if you have an extreme emergency, for example shock, the renal blood flow can be reduced to very low levels so that it can raise the pressure to and as a result the blood flow to other organs like the brain and the heart. However, keep in mind that the kidney is using also a lot of oxygen, so you cannot keep the blood flow low for too long, because renal damage will be the end result, and if that happens, then you lost the game of using it as a hemodynamic reserve. Finally, the renal blood flow has to be controlled in order to deliver sufficient oxygen and nutrients to the kidneys, but we all have to agree with 1 liter per minute flow rate, this oxygen and nutrients will usually be very plentiful. Okay, so it's not something that the kidney is going to be starved of. Now, the regulation of the renal blood flow is a very peculiar subject, a very difficult subject for a lot of people, but it can be done through 4 basic ways, some of them more complex than others. The first one is autoregulation. What it means is the renal blood flow regulates itself. And the regulation in this case is happening at the afferent arterial level. And there are two types of regulation. One is the autoregulation. Well, we are using really the same term here, but let's call it intrinsic autoregulation, so that we distinguish it from the other type, which is the myogenic autoregulation. We're going to talk about it in a minute, about what each one of these two means. The second way of regulating the renal blood flow is through the so-called tubulo-glomerular feedback mechanism. Okay? Now, the name itself means that there is a feedback mechanism between the tubules, which is the proximal tubule, the distal tubule, and the collecting tubules, okay, and the glomerular apparatus. What's the glomerular apparatus? Is the tuft of capillaries that are inside the Bowman's capsule. So between these two organs, these two different parts of the nephron, there is some kind of a feedback, again, Farther down the line, we're going to get more detailed about this. Then we have the so-called renin-angiotensin system. Renin is one of the most powerful, and angiotensin, there are some of the most powerful vasoconstrictors known, okay? And obviously they have a role to play here because through vasoconstriction you can control flow, that's number one. But number two is because the basis of them is actually the kidney. Don't forget that renin and renal have the same root in Latin, and it means kidney, right? So it means it is a system that is based in the kidney. Finally, you have other vasoactive agents in the kidney, and I will stop at two kinds. One is the vasoconstrictors, which is the endothelin, and the name means that it is produced by the endothelium, which is the lining of the blood vessels, and 8-arginine vasopressin, or AVP. Okay? The other class is the vasodilators, and over here we have the prostaglandins, nitric oxide, and the natriuretic peptides. Remember, up to now we have talked about one natriuretic peptide, which is the atrial natriuretic factor, okay, or peptide. But there are others. So, the intrinsic autoregulation occurs at a mean arterial pressure between 70 and 210 millimeters of mercury. And it keeps the blood flow and therefore the filtration rate nearly constant or always constant despite any pressure changes. On the other hand, during myogenic autoregulation, there we have a different kind of regulation whereby there are stretch-activated ion channels in the vascular smooth muscle, and when those are stretched, they are activated, and they allow the calcium ions to enter the cells and produce contraction. So this is why this is called myogenic, and this is in effect different than the intrinsic, which has to do with the input and the output balancing together. Okay? Now, the second one, if you recall, we talked about the tubuloglomerular feedback. And tubuloglomerular feedback occurs between the macula densa. Now, I'm sure that you have talked about it in your histology class, what the macula densa is. But to give you an example, if you look at the top picture there, which you have already seen, you see that there is an afferent arteriole and an efferent arteriole coming out of the capsule of Bowman. Well, they are, let me go on the board and draw it for you so you understand it a little bit better. The Bowman capsule, although in our books we stretch it so that we can show, the nephron, not just the Bowman capsule. We stretch everything so we can see what its part is separately, but the Bowman capsule continues on to the proximal convoluted tubule, and from that point on goes on to the loop of Henle, onto the distal tubule, and this is what we normally have in the books, okay? And normally that's how we put the efferent and afferent arterioles. Well, actually, in order to make sense out of this and what I'm telling you about the macula densa, you have to figure out that you split everything right in the middle of this Henle's loop and transfer this on top here, so that in effect it's going to look like this. Okay, if we had turned this around like this way, so that it comes one on top of the other, then this part would come and pass through the point where the two arterioles come into the nephron. Is that understood?
Yes.
In other words, if you look at my fist, can I zoom in? If you look at my fist, okay, and you consider that this is the nephron, and if you look at my two fingers and you say one's the efferent and one's the efferent, then all the tubing that comes out of here eventually it will come and it will come and pass like this between the two arterioles. The distal tubule will pass right through here. Okay? So in between the distal tubule and the two arterioles there is a small triangular space. And that triangular space, which is this one here, okay? This is the incoming. This is the outgoing blood. And right in between comes the tubule, okay? Over here in this, between these three vessels, two of them are blood vessels and one of them is carrying the urine filtrate. Well, that's where the macula densa is. And if you look at the bottom slide, which I'm sure that you have seen at the bottom slide, which I'm sure you have seen in your histology class, you'll see there in the center of it you see the glomerulus, and on top of it you see this triangular space which is empty in the middle. Okay? Can you discern that? Can you see that? Well, that is the distal tubule. It's empty in the middle because that's where the urine passes from. And below it, it has this thickening of the cells. You see the cells are a little bit thicker than the other cells on the other two sides of the triangle. Okay? And that's where the macula densa is. If you compare this with the one on top, you are going to make out how the macula densa looks. Is it clear or not?
Clear.
Okay. Now, when fluid delivery, that is how much blood goes through to the nephron, or the sodium chloride transport, in that particular part, in the macula densa, they increase, as in the case of the increased glomerular filtration. Then there are increases in the cell sodium and calcium. And this results into the release of arachidonic acid metabolites and adenosine. This in turn, they act on the vascular smooth muscle cells of both the afferent arteriole, causing contraction, and sometimes on the efferent, and they cause contraction, and this way they reduce the blood flow. Okay? On the other hand, if we reduce the sodium chloride that goes to the macula densa or the blood volume, then the opposite is going to happen. Arachidonic acid metabolites are going to decrease, adenosine is not going to be released, and you're going to have dilation. So, you see, this is a mechanism that works right at the local area. One cell releases, the next cell receives the particular signal, which is in this case arachidonic metabolites and adenosine. Now, is it clear how this tubuloglomerular mechanism works? Yeah. Okay, this is very good. Please look at those slides on your own, because it's really important that you understand how these things are built. Well, this is another example where I found a nice picture to show you of the same thing, of the apparatus, okay? And you have the glomerulus all the way down, but you see there the macula densa and this triangular space on top of it. Okay? Now, the feedback occurs between the macula densa cells and the cells of the afferent arteriole, which is called juxtaglomerular apparatus. That means attached to the glomerulus, apparatus that is attached to the glomerulus. And the third system that we are going to talk about is the renin-angiotensin system. And I have to tell you here that again we are at the same place in the kidneys. Renin is produced at the granular cells of the afferent arterioles. So all these mechanisms are literally one very close to the other anatomically. It is released in response to decreases in renal perfusion pressure, and normally this is done by sympathetic stimulation through the renal nerves. And also it's done by reduced flow and transport in the macula densa cells. In other words, the macula densa is producing, in response to the stimulation, the same metabolites that it produces can also trigger the renin-angiotensin system. Now, here's a diagram. I don't know if you can see it very well, okay? But it gives you the course of events that happen throughout the body when you have an emergency. What is the emergency? In this case, this is decrease in blood pressure. Well, there are small arrows, either red or green or blue, that show you what happens. All right? So if you have a decrease in blood pressure, you're going to have a decrease in glomerular filtration rate. This means you're going to have a decrease in sodium in the convoluted tubes, and this, of course, is going to bring forth the mechanism of the macula densa, okay, which in turn is going to to secrete renin in the plasma. Renin is going to act on the angiotensin 1, angiotensin 2 system, and because of that you are going to have contraction of blood vessels and increased in blood pressure. So this is one way you can go through. The other way is that decrease in the blood pressure itself acts on the mechanoreceptors of the juxtaglomerular cells. Remember what we said about the stretch receptors, right? Again, have secretion of renin in plasma, action on the angiotensin system, and this also can bring constriction of the blood vessels. Although in this case, angiotensin can also create something else. It also can act on the aldosterone system, and this will increase the uptake of sodium and water. So all these things are separate mechanisms. We are going to go in through them in a minute, but they all together show you how the renal blood flow is regulated. Now, renin in particular is a protease. Protease means that it is a protein that cleaves proteins. It is an enzyme that cleaves proteins. Now, that protein reacts with angiotensinogen, which is a precursor to angiotensin, and angiotensin 1, which is the first precursor that we meet here, has 10 amino acids, and it is converted to angiotensin 2 by an endothelial angiotensin converting enzyme called, or ACE, not called, that's how it's called. It's called angiotensin converting enzyme, and we abbreviate this as ACE. Now, ACE is something that you are going to find out a lot in your lives as doctors, because most people nowadays, when they are over 45, they take ACE inhibitors to keep their blood pressure controlled and their heart working fine. Now, angiotensin 2 is a very potent vasoconstrictor, and it acts preferentially. It acts preferentially in the afferent arteriole, reducing the blood flow, but at the same time maintaining or even increasing the filtration rate. Okay? How does it do that? Because of course it constricts the blood flow, but it also raises, if you constrict all the vessels in the body, what will happen to the volume? The volume cannot be compressed. Therefore the pressure will rise, correct? So you may on one hand reduce the flow, and the other you increase the pressure, so you could play with these two things, one against the other, and manage to maintain or even increase filtration rate. Angiotensin 2 also acts on the adrenal cortex, and there it induces the production of aldosterone, which is a hormone that stimulates sodium reabsorption where? In the distal tubule. And in the proximal tubule, it stimulates the reabsorption of sodium bicarbonate. So between these actions, you can see that it's with all of them working in parallel and complementing each other, it is quite easy for the kidney to maintain the perfusion rate that it has at a good level. Now, of course, there are always, there is always a problem in paradise, and at higher plasma levels, angiotensin 2 can cause the mesangial cells to contract. And the mesangial cells is the cells of the parenchyma of the kidney, and it's called mesangial because they are between vessels, right? And these are cells that can contract, and when they contract, this increases the pressure on the vessels, and it decreases filtration. Also, angiotensin 2 can cause a generalized vasoconstriction, which not only acts on the afferent arteriole, but also on the efferent arteriole. Now, this vasoconstriction helps maintain the central arterial pressure high, but this is done at the sacrifice of renal blood flow. Remember what we said about the emergency response mechanisms a few slides back, that if need be, there is a lot of reserve that the kidney has in order to keep the body blood flow constant. Well, this is one of the ways that it does it. Actually, it is the main way to do it. Now, last but not least, we talked about other vasoconstrictors and vasodilators, other vasoactive agents, as we say in the kidney. One is endothelin, and we said where it's produced, and the other one is 8-arginine vasopressin or AVP. They are both used to reduce the renal blood flow. AVP, what it does, it acts mostly on the medullary blood flow, and also it acts on the mesangial cells to reduce filtration, something like angiotensin II does. In terms of the vasodilators, there is prostaglandins, nitric oxide, and the natriuretic peptides that we said, and these are there to counteract and limit the effect of the vasoconstrictors. In fact, today we suspect, or we are fairly sure by now, that it is their absence or their blockade which are the causes of hypertension, the causes of profound renal vasoconstriction that sometimes hits people, and reduced filtration. Any questions up to this point? I just have to make one small comment here. Difficult as these things may sound, they really are not that difficult. And it is really important for you to learn these particular mechanisms because you are going to need them. These are questions you're going to find in front of you, not only in your exams, but mainly in your practice, irrespective of what branch of medicine you're going to follow, what specialty. Now, let's go to glomerular filtration for a minute. And say that the initial step in glomerular filtration is the formation of urine. Okay, rather the first step in the formation of urine is the production of the filtrate in the glomerulus. Remember what we said. This is bringing your mind, if you don't mind, the example of the refinery. The first thing you have to do is filter what you have. And as in the case of the capillary exchange, and I promised you last time that when we talk about the kidneys, we are going to talk about capillaries also. Good. Can you pass on for me?
I can try.
All right.
George, you have to come here to have them.
Yeah, I know. Okay. Now, as in what?
Nick will get couple for you.
Okay. Well, I suggest you eat them all because they'll be stale by the time I come. We'll get new ones then.
We'll get them after you come here.
Okay. Now.
It's really good, George. It's really good. Very yummy. And you can only get it in Bulgaria.
Guys, I am really tickled pink that you think of me in such nice terms. I promise you I'll come over.
We miss you, George. Come on.
Okay, now let's start again talking about the formation of urine. Okay, the first step of producing urine is to produce a plasma filtrate within the glomerulus. We said how it is done, but I promised you last time, I said, that when we come to this point, we are going to discuss side by side what happens in the capillary beds, in the somatic capillary beds, the capillary beds across the body, and what happens in the capillary beds in the kidney. And I told you then that I prefer to discuss these two things together rather than separately. But again, this is my own choice, and you can always look at them as two separate things. They are separate, but on the other hand, one is telling a story for the other. So, the driving forces, whenever you have exchange across capillaries, across capillary walls in effect, are the hydrostatic pressure and the osmotic pressure gradients between the internal and the external part of the capillary, and in this case between the glomerular capillaries and the Bowman space. Okay? There are basically across our body three types of capillary walls. One is called the continuous wall.